This is just a short tutorial about fermentation. Now, fermentation is a very common process, but it's fundamentally different than respiration. In respiration, that's where electrons are passed to an external electron acceptor such as oxygen, and that's what we do when we breathe. We're using oxygen as our electron acceptor. And during fermentation, part of the original substrate, and the one we'll talk about today is glucose, is used to regenerate oxidized cellular electron carriers such as NAD, and you'll see this in the next slide. So that means that part of the original substrate ends up being what's considered sort of a waste product. Well, let's look at a couple of different examples of fermentations. These are 5 different fermentations. They all start with glucose, which is a 6-carbon carbohydrate. And in the process, glucose is broken down to pyruvic acid. This is a process that's well known. It's called glycolysis, and it's going on in your body all the time. Now, during this biochemical reaction from glucose to pyruvic acid, an important molecule known as NAD, which is an electron carrier, will play a major role in this, and in the process, NADH is generated. Now, the cell needs to get rid of that NADH and regenerate NAD, and that's what's shown in the reaction down here. Now, normally in the body, NADH reacts with oxygen using some enzymes, and that's how NAD is regenerated, because oxygen is the electron acceptor in this case. But in these fermentations, there is no oxygen. So NADH, in order to get back to NAD, needs to be involved in these steps here. And in each case, these organisms each handle pyruvic acid in a different way. So, for example, for Propionibacterium, in order to regenerate NAD in its reaction, it takes pyruvic acid and makes propionic acid and carbon dioxide. And that reaction is very important because it's how Swiss cheese is made. In milk, the Propionibacterium is growing. It makes propionic acid, which gives the Swiss cheese its flavor, and the carbon dioxide gives those nice holes. If we're looking at pyruvic acid going to lactic acid, that's a reaction that organisms such as lactobacillus and streptococcus use, and in the process of doing that in milk, they will produce yogurt. Now, if we look at the situation that involves Saccharomyces cerevisiae, it regenerates NAD by converting its pyruvic acid to carbon dioxide and ethanol. And when it does that with grape juice, for example, it produces wine because it has the ethanol for wine and the carbonation that we see in wine also. And of course, given a different substrate with grains, then it would produce beer. Clostridium is used industrially, and it converts pyruvic acid to acetone or isopropanol, giving us nail polish remover and rubbing alcohol. And finally, Escherichia and Acetobacter can produce acetic acid from pyruvic acid, again regenerating NAD, and that is known as vinegar. Here's a slightly more detailed diagram showing the breakdown of these products, and it's also comparing this fermentation, which, by the way, in some cases is called an anaerobic metabolism because there is no oxygen. This happens in your muscle when you are using glucose, but you're not getting enough oxygen to the muscles, and you get a little bit of buildup of lactic acid, which can make your muscles sore, and that's going to happen in some cases. We're going to be in the laboratory looking at what happens when yeast takes glucose and makes ethanol. And here are what yeast cells look like under the microscope. They're color enhanced here, but you'll notice that these are yeasts that are budding. Notice that glucose again gets converted in several steps to pyruvic acid, which is a 3-carbon. This goes from a 6-carbon to a 3-carbon compound. And then carbon dioxide is lost. That's where that carbonation comes from. If you stop the reaction at this point early on in the game, you'll get a lot of carbonation, but you won't actually go to the stage where you make all this ethanol because there's still enough oxygen in a typical yeast culture to allow the cells to grow. However, as you saw in the previous slide, the reason that pyruvate goes to acetaldehyde and eventually to ethanol is to regenerate the NAD from the NADH. And that's because we only have a certain amount of NAD in the cells, and it needs to be regenerated at all times. As we saw in the earlier slide, the lactic acid bacteria are involved in the production of yogurt, sauerkraut, and products like kimchi. And again, they take glucose, run it through the process known as glycolysis, which you'll learn more about in both microbiology and biochemistry, to give us pyruvate. In the process, this is important for the organism because it gives them ATP for energy, but the pyruvate ultimately gets converted to 2 lactic acids, lactate, or also known as lactic acid, and again, that's to regenerate the NAD for the cell. Well, why do we care about lactic acid production? Well, historically, of course, the reason for this is that without any refrigeration, if you had milk, it would go sour very quickly. So naturally, making yogurt or kefir, which is a product that's similar to yogurt, was really a natural way to preserve the milk. So what happens if you have milk and you add these lactic acid bacteria, they're going to grow in that milk because there's a good source of carbohydrate and protein in there, and they will lower the pH so that in essence you have a sour milk product. Other bacteria and yeast will not be able to grow very well in that, so you have naturally preserved it. Plus, you're now consuming the yogurt that has the nutrients of milk, plus all of the microbes that have grown in there, which are good for the body. And as I said, this is a great way to preserve the milk as well as acquire a source of microbes that are going to be good for you. And the reason that this pH gets lowered, of course, is because of the lactic acid that the bacteria produce as they grow.